you all for the kind introduction and for the invitation. Yeah, so today I'll talk about one of our work they call TxGNN, which is the foundation model for drug repurposing. And this is a joint work done with amazing group of people, Paya, Qianwen, Jay, Akio, Yuri, Jirish, Ben, Niels, and Marinko. Yeah, so today's talk will be divided into three parts. So first, I'll just briefly talk about the state of AI drug repurposing for rare disease, and then I'll go into our method and talk about why the motivation and why we're developing TxGNN, and then I'll focus a lot of time on evaluation in the wild and also deployment as well. So as probably a lot of people know, like there's thousands of diseases out there, rare diseases out there, impacting millions of patients collectively, but only very few of them have treatments. Part of the reason is drug discovery is just so slow and costly. It takes more than 10 years, more than billions of dollars to develop a drug from scratch and get it to market. And also most of the rare diseases actually do not have active drug programs. Uh, and there's a lot of work trying to do drug repurposing for rare disease because it has all kinds of different benefits, right? Because for the repurposing, usually the drug profile is like very familiar. We know we have tons of preclinical knowledge about the drug. We have and also if it's repurposed, then you don't need to go through like phase one phase one trials, right? Because it's already safe. And also it has basically faster clinical translation. So it's definitely a suitable path, especially for rare disease. And historically, repurposing is usually found by serendipity. For example, the most famous example is the sildenafil, which is originally used is for high blood pressure, and then you have later like a completely different indication as well. And our question is, we can do it systematically. And the motivation of why, how do we do it systematically is actually the motivation is like biological system is a huge graph. What I mean is like there's protein, there's non-coding there's small compound, there's neural diseases, EHR, and everything is like interconnected. So you can model it as like huge graph. And a disease is basically, if we are formulating under this network phenomena, is just like a module of like a subgraph from this massive graph. And a drug treating a disease is basically perturbing the local biological system of a disease such that it creates some therapeutic effects. So this can naturally gives us a graph machine learning formulation. Basically, we can just ground the model to a knowledge network about disease mechanism and treatment effect. Then we can use graph machine learning trained on these known indications, and then we can potentially infer on unseen indications. So in order to use this graph machine learning formulation, we first need to create such a graph. So earlier in 2023, we created a prime KG, which is a therapeutic-centered knowledge graph, which capture majority of the genes, diseases, pathways, and it's designed for rare diseases. And it covers around 4 million connections and 100K nodes. And then AI has definitely shown promises in drug repurposing in the past few years, but we are really thinking about why is there no existing AI-made drug repurposing delivered to patient yet. So we're developing a method to try to really evaluate what's preventing AI from being useful, what's the issue with the current evaluation setup for AI-driven drug repurposing, and then we propose a new method for it. So we actually very interestingly find that actually the biggest issue is actually the evaluation setup for the existing AI models is not reflecting the real world challenges. Uh, currently, people only evaluate on drug and treatments with a large number of like existing treatment for diseases and also strong molecular understanding, right? But so in this kind of settings, if you just use a simple graph neural network architecture, you're able to get like 0.9 AUPRC, which is what the other people have reported as well. But the issue is like these are not for the rare diseases because for the rare diseases, there's no existing treatments for disease, and also there's very limited molecular understanding for disease, right? Um, uh, and when we evaluate on this setting, we see that the performance should dramatically drop, and then But this is actually what we want to really evaluate. Um, so part of the reason why it's so easy for this evaluation is because you can basically, if this disease is already have many treatments, then you can just do some like drug-drug similarities, and you can just predict it very, very well. So our goal is I want to close this gap, right? We want to enrich the disease embeddings such that we can close this gap. Because right now, if you look at the graph of this disease node, it's not connected to much other nodes. So the embedding of the disease is actually really bad. So we need a way to kind of systematically enrich disease embedding. So here we introduce two techniques. One is metric learning, and the other is self-supervised pre-training. So what does this mean? So for machine learning, it's basically the intuition is because biological diseases in a network medicine domain, there's a local hypothesis, which means that diseases that share some similar molecular mechanisms, have similar profile in the biological processes, molecular functions, and so on and so on. So we're thinking maybe we can just borrow from the other diseases, learning from similar diseases to enrich the disease embedding. And we're basically using a small set of links as anchors to find the similar diseases and expand the disease modules. And this is basically means we want to help diseases to help diseases in need. Then the open question is like, how do we identify such similar diseases? And then is how do you leverage the disease similarities in the graph neural networks? And we first use a knowledge graph derived disease similarities. We actually experiment different techniques. Then we realized that this knowledge graph, this derived disease similarity is very useful. Basically, we can do like common neighbors, or we can just count the frequency of co-visited nodes in a multi-hop diffusion. And then we can just create a profile for the disease, and then we can measure how similar is the disease. And we find that using this disease similarity, we were able to capture the disease landscape relation really well. For example, as the score is higher, the diseases pair is actually more similar, right? So pick disease and Alzheimer, they are very similar. And the majority of the diseases are not that similar to each other. So it have very low scores. Then how do we enrich the embedding in a graph neural network? What we do is we augment the embedding by doing some simple aggregations. Right? Is this like after we use the signature that generated from the first part, then we can just merge this embedding through some aggregation mechanisms. And if you think about it in machine learning terms, what it's doing is basically doing some graph rewiring, where basically for this is target disease, if you just use the two-hop local neighborhood, the embedding is really bad because of the issue that I mentioned before. But if you use this kind of techniques, it's actually theoretically doing this long-range selective aggregation guided by some domain priors. And this domain prior is basically the disease similarity that we measured. And then so that was strategy one. And then for strategy two, we're thinking we can use like self-supervised learning over graphs because in a knowledge graph there's so much information in there. And if you just directly train on disease drug in relationships, it's not sufficient. Potentially there's a lot of insights in the other type of relation as well. So what we did is we use a pre-training of the disease of the knowledge graph. And this pre-training is done by doing edge perturbation or node dropping or random walk sampling, and then we can just reconstruct it as the learning tasks. So in summary, we propose a new method called called TxGNN, which is a graph foundation model for drug repurposing. And to evaluate it, we perform a large number of evaluations. So first, we evaluate on the previous evaluation setup. We observe that TxGNN has similar performance as the other baselines. But when we evaluate it on the real-world settings where the disease has not much training drugs, and then we're able to see TxGNN has the best performance compared to all the other baselines. And we further evaluate a more challenging setting where we mask the entire disease areas from the training set for the target disease of interest. And we find that our TxGNN still has the best performance across a wide range of disease areas like adrenal gland, autoimmune disease, neurodegenerative disease, metabolics, and cardiovascular. Cool. So that's a quick overview of the method and also the kind of some kind of basic evaluation that we did. And then we'll delve into the evaluation in the wild and then also deployment for clinicians because we still feel like this in silico evaluation has a lot of assumptions. So we want to use real world evaluations to see how it is really useful. So we partner with Mount Sinai, which they have a massive number of EHR datasets. And what we did is to really validate the predictions made from TxGNN, which is the drug disease relationships. We need to have some proxy of how good is the drug, the novel predictions in the real world evidence, right? So what we did is that for the Mount Sinai EHR data, they have curated, basically processed the EHR data, and then they send us a spreadsheet which is curated a cohort of 480 diseases with larger than one patients and 1,290 disease drugs with larger than 10 patients. And for these, we have more than 1 million patients with at least one drug and at least one disease belonging to them. And what we did is we just computed log odds ratio per drug disease pair. Right? If the log odds ratio is high, that means that this drug and disease is co-occur much more frequently compared to like the other drug disease pair. The motivation is like one because it's impossible to do like clinical trials for each prediction that we made from TxGN. But we know that there's a lot of kind of clinicians based on their knowledges, they just prescribe this off-label prescriptions for the drug. So we're thinking maybe this co-occurrence log odds ratio is basically measuring this kind of a off-label prescription, which is aligned with the clinician how this drug could be treated with disease. And we're using that as a proxy for the drug for validating the novel predictions TxGNN made. So we did some feasibility benchmark where we indeed find that the FDA-approved drug disease pair indeed have a much higher log odds ratio. So this means that if our novel predictions made by the TxGNN has high log odds ratio, it's more likely that the clinician think it's like a useful drug for the disease. So we do find that the top one predictions made by TxGNN has a significantly higher enrichment compared to the top 5%, and also bottom 50% of the predictions. And this is also but it's definitely far away from the approved indications because these are novel predictions made by the model. These are not the existing drugs. But we find that it's basically aligned with the off-label prescription patterns of the clinicians. And then we find there's two examples of novel predictions. One is like this astrocytoma, and another is Wilson disease. And we find that, so for example, the TxGNN identified these drugs, and the log odds ratio is really high. And also similarly, the Xerox, also TxGNN have very high predictions, and log odds ratio is also really high. And they all both have like a literature evidence as well. But we still find it's probably not super actionable. So we did is like we realized that the prediction alone is not trustworthy because it still has errors. So we need to have the predictions lend themselves to actionable hypotheses. Where we need to guide downstream experiments and clinical inquiries. So what we did is we grounded by a knowledge graph. We can basically use exam and visualize the rationale for model predictions using graph explainability approaches. So basically, back then there's a method called graph mask, which is published at iClear, and we're adapting it to our settings, where we're able to basically graph mask. Initially, it's designed for like a homogeneous graph, but right now we have a heterogeneous graph, and so basically the intuition is that based on the node embeddings, it will feed into a weight, with a sigmoid layer. And if you want to, if the score is larger than 0.5, you just keep it. And if it's not, we just mask it. And then this automatic layer, it will automatically update the weight here as it learn. The goal is to mask as much edges as possible while keeping the loss similar. So this way, you will just have the leftover. You basically generate a score for each edge, and when the score is higher, it means the edge is more important for the predictions. And we created a TxGNN Explorer available at txgnn.org. And this is readily available for everyone to use, where you can select the disease, you can select the drugs, and then it will pop up the explanation generated by the TxGNN. And the user can basically select the drugs, can also explore the path explanations. And the user can hide, unhide, collapse, and expand the group of explanation path based on the meta path. And we did a user-centric studies to really see if the XAI is useful. So what we did is we have 16 pairs, 12 of them are correct, 4 of them are wrong. And then we give the predictions and also accompany with different, if you get no XAI, with different kind of XAI as well. And for every task, each user is asked whether the prediction is correct and also the confidence level in the 5-point scale. And we basically recruit 12 people with 5 physicians, 5 clinical researchers, and 2 pharmacists. And this is the user study interface that we did. We can basically see that this AI predicts that the disease can be treated by the drug, and this is give a confidence score of this for its prediction. And also you can also adjust the edge threshold to see the visualization, and also you can select your decision, rate your confidence level, and so on. And we find that interestingly, using XAI dramatically improved the adoption of AI for the clinicians. It improves the accuracy and confidence. For example, the accuracy of ours is now zero 0.8, compared to the baseline is just 0.5. And also increase the confidence by more than by a lot when with statistical significance as well. And then, we also have a more qualitative evaluation for our platform, and we find that the users and the clinicians find it, useful and helpful, and the XAI is really useful for how understand the predictive drugs, and also is willing to use this AI. And another interesting thing is that we actually measure after the use of XAI, what is the performance of human accuracy. And we find that the human accuracy is actually 0.8, so which is higher than the model accuracy. But without XAI, the human accuracy is only just 54%. So this means that the XAI really helps improve trust and thus improve adoptions. And then when there's no XAI being shown, the human user just mistrust AI predictions and they use their own judgment, and then it's more likely to fail. And here is some real world examples on the path and also the reasonings. For example, this is the explainer, the mechanisms, and and this is for this ED syndrome, which is also a rare disease. And our agent is able to our platform is able to recommend this drug as the top one. And then there's also the rationale why this drug could be treated the disease and the reasonings behind it as well. Cool. So this is a very quick summary of our platform, you can. So this is it's published at Nature Medicine, I think 2 years ago, and then it's also covered by different journals. And feel free to read more about it. There's much more details in the paper as well.